We're HHMI-Brown Scholars.
My name's Charlotte Kim. I'm from Los Angeles, and I'm a rising junior studying biology.
I'm Steve McShane from Detroit, Michigan, and I'm also a rising junior studying chemical biology. And our experiment is on the dose-dependent inhibition of gap junctions and P-glycoprotein.
Hi, I'm Jieyi. I'm a rising sophomore studying history and biology.
Hi, my name is Nguyen, and I'm a rising second year studying education studies and biology. And the name of our project is the durations of effect of gap junction inhibiting drugs.
Hi, I'm David. I'm a rising sophomore from Arlington, Virginia, and I'm going to major in chemical biology.
Hello, my name is Lucia Hernandez. I'm a rising junior. I'm going to be majoring in biomedical engineering, and our project is gap junctions and chimeric spheroids.
Hi, my name is Nimesha Gerlus, and I'm a freshman from Springfield, Massachusetts. I'm concentrating in cognitive neuroscience.
Hi, my name is Kadota. I'm a rising junior concentrating in biology, and our project is synergistic effects of ABC transporters and chimeric steroids.
Alright, so what is HHMI? HHMI is an intensive 10-week summer program done in collaboration between Brown and the Howard Hughes Medical Institute. It's a program that's designed to help engage rising second and third year students in scientific research. It helps place undergraduates in the hands of faculty and the scientific community here. I, for one, know it's hard to approach professors during your lecture classes, so being able to see them in different settings is quite helpful. The program itself has about 32 students placed in 4 groups of 8, and our group specifically works with Dr. Morgan. We are the drug discovery group, and this summer we've been working with the graduate students to help design and run our own experiments.
The format and timeline for the summer was 2 parts. The first 2 weeks we worked on basic lab techniques from how to work in a biosafety cabinet and the basics of aseptic technique to cell culture maintenance. Then we learned how to use the Morgan Lab's 3D technology to make agarose gels and form 3D tissues. The final phase was learning microscopy and using fluorescent imaging. All of these skills were vital for our individual projects, so that the remaining 8 weeks were spent working with our graduate students to develop and run our experiments. All of this would culminate in a poster symposium at the end of the summer.
Hi, I'm Charlotte, and I'll explain what our 3D technology is about. So the 2 research models that are commonly used in other labs are in vitro animal models and 2D tissue culturing in vitro. But what are some problems with animal testing? For one, it can lead to ethical concerns. And it doesn't always work, and when animal models fail to simulate the human body, this can be dangerous for us too. An example of this happened in 2006 when a German company tested their first immunotherapy drug called TGN-1412. This drug was deemed safe for animals, but it nearly killed all of the human volunteers, despite being administered in much smaller dosages on the humans. Last year, Forbes magazine noted that companies can spend billions of dollars on each new drug, so such failures are also very unsustainable. So it seems that 2D tissue culturing might solve some of these problems. It's cheaper, and it allows scientists to use human cell lines and test more conditions on many more subjects, which can save time. But of course, our own tissues don't exist as simple monolayers of cells, and this may affect molecular transport and diffusion in the human body. So the technology that our group uses is Dr. Jeffrey Morgan's 3D Petri dish, which lets us culture cells in 3-dimensional shapes, particularly spheres. To do this, we insert liquid agarose solution into these green molds and wait a few seconds for the agarose to cool and harden. Then we're left with a gel that can be equilibrated in media. We seed the gel with monodispersed cells, then the cells settle by gravity into the microscopic wells. The gels are non-adhesive to the cells, allowing them to self-assemble into spheres without the need for building any scaffolds. A great advantage of this technology is that we can control the size of our microtissues simply by changing the seeding density. The Morgan Lab also investigates tissues in more complex shapes, such as rods, toroids, and honeycombs.
Hello, my name is Lucia. I will be talking about the background of all our projects. First, I will talk about gap junctions. Gap junctions are small channels between cells. They help with intracellular communication. Using our 3D cell model, we have a better representation of gap junctions. We will also be studying PGP, which is P-glycoprotein. This is an efflux pump with a wide range of substrates. It pumps out toxic molecules such as xenobiotics from cells. Tumors upregulate the expression of PGP pumps. That means it drastically increases the expression of PG pumps, which then pumps out the drugs that want to enter the cell. This plays a significant role in the multi-drug resistance. We will be using calcine AM as our model drug for a dual assay of gap junction inhibition and PGP inhibition. Calcine AM is non-fluorescent and can passively diffuse across a lipid bilayer of a cell. However, once it enters a cell, it is cleaved by esterases and becomes fluorescent calcine, which is then trapped inside the cell. Fluorescent calcine can only diffuse via gap junctions. So in our 3D model, the calcine travels from the outer to the center. We can quantify gap junction activity by measuring the total calcine distribution in a spheroid. As you can see in the diagram, calcine AM starts in the outside of the spheroid, it enters through the outer cell layer, and once it enters the outer cell layer, it is cleaved by esterases, becomes fluorescent calcine, and then diffuses via gap junctions towards the core of the spheroid. If we add gap junction inhibiting drugs, then the calcine no longer can travel towards the core of the spheroid. A common gap junction inhibiting drug is carbonoxylin, also known as CBX. We also quantify PGP activity by the total amount of calcine in a spheroid. PGP, or P-glycoprotein, pumps out calcine AM. As mentioned, we will be investigating gap junction and PGP activity function with our model drug calcine. But how do we analyze the spheroid assembly? We homogeneously label the spheroid with cell tracker red. This helps us identify the size and amount of cells in a spheroid for further data analysis. Once we have gone through the process of seeding our spheroids into the 3D Petri dishes, adding model drugs and inhibiting drugs, we take time-lapse images. As you can see, over time, the calcine in the cells becomes brighter. Specifically in this time-lapse, you can see that the outer rim of the spheroids is much brighter than the center, indicating that we have used a gap junction inhibiting drug. Next, I will discuss how we perform our data analysis. We quantify PGP activity by calculating total calcine in a spheroid. As seen in the equation, first we take the total raw fluorescence of a spheroid, subtract the background fluorescence, and then normalize by the surface area. As you can see in the graph below, calcine fluorescence increases over time. However, when you add a PGP inhibitor, the calcine is no longer pumped out by the PGP, and thus more calcine is accumulated inside the spheroid, which increases the total fluorescence. Gap junction activity equals calcine distribution in the spheroid. We measure the center fluorescence and divide by the total fluorescence in the outer rim. We do this to quantify if calcine diffused into the core of the spheroid. Below is an example of a gap junction inhibiting drug being used. To the right, you can see that the fluorescence is much brighter on the outer rim versus the center. And to the left, you see when there is no gap junction inhibition, the calcine diffuses throughout the whole spheroid. The complete equation is then taking that ratio and dividing by the cell tracker red labeled cells. This is to normalize by the different sizes spheroids and different amount of cells. The graph below is an example of gap junction inhibition quantification. As you can see, if there is a gap junction inhibitor present, the ratio is much lower. There is more calcine on the outside because there is gap junction inhibition, which means that the calcine is stuck on the outer rim and cannot diffuse into the center of the spheroid.
Hi, I'm Steven, and I'll be introducing our first set of projects. For our work this summer, our team of 8 split into 2 main subgroups of 4 each. And the first subgroup, consisting of myself, Charlotte, Mary, and Jieyi, looked at developing a system for drug characterization of gap junction inhibitors. We focused on what we term chemicals of commerce, which are things like pesticides, pharmaceuticals, and other chemicals that are in our day-to-day environments, specifically with the goal of exploring any potential toxicity of these. And we did this in 2 main ways, exploring dose-response curves and examining duration of effect of these chemicals. And the chemicals that we initially studied were carbonoxylone and mefloquine, which are 2 well-known and well-characterized gap junction inhibitors in order to better refine our assay. And then we began looking at what the chemicals of commerce that I mentioned, using the pesticide lindane. Charlotte and I focused on the dose-response portion of this project. And our experimental design for this is similar to the other 4 groups that you'll see today. We began by homogeneously labeling our cells using cell tracker red. After that, we passaged them into our microtissue and allowed the spheroids to self-assemble overnight. We came back the next day after waiting 24 hours and pre-treated the spheroids with our gap junction inhibitor in question. After waiting 5 hours to allow the drug to diffuse through the entire spheroid, we added our calcine AM and then began to image for 2 to 3 hours. As previously characterized in the Morgan lab, our results showed that carboxylone is both a PGP inhibitor and a gap junction inhibitor. As you can see here in our graph of surface area normalized fluorescence over time, carboxylone shows higher total fluorescent uptake as the dosage increases, indicating that there's more calcine diffusing into the cell and thus more PGP inhibition. Our results were different with lindane and mefloquine, which showed no PGP activity, as you can see here with our positive control of 100 micromolar CBX, which shows much higher fluorescence than any of our lindane or mefloquine doses, indicating that PGP was not affected at all with either of these drugs.
Hi, I'm Jieyi, and I'm going to talk about our individual project, which is on the duration of effective gap junction inhibiting drugs. When determining how long a drug lasts, current drug testing only focuses on how long the drug remains in the bloodstream. However, a drug might still have an effect after it has cleared the body. Duration of effect describes how long a drug has an effect on the system, regardless of its measurable presence. So our experimental design is very similar to what has been described previously, except that in order to examine the duration of effect of gap junction inhibiting drugs, we wash the drug out after 5 or 24 hours of treatment time. We then add the fluorescent calcine dye and take time-lapse images of the spheroids over the course of 2 hours to monitor PGP and gap junction inhibition over time. Here you can see the results for a dual assay with 5-hour carboxylone treatment. We have 4 conditions tested: a no-drug control, a 4-hour washout in which the spheroids were treated with drug for 4 hours and then washed out and left without drug for 1 hour before imaging, a 5-hour washout condition in which the spheroids were treated for 5 hours, washed out, and imaged immediately, and a 5-hour continuous condition in which the drug is never washed out and the spheroids are continually exposed through the imaging. In the PGP inhibition graph, we have total fluorescence on the y-axis over time on the x-axis. Higher total fluorescence indicates more PGP inhibition because, as previously explained, if PGP is inhibited, it can no longer pump calcine out of the spheroid, and so the calcine is trapped in the spheroid and the fluorescence increases. We can see here that the no-drug, 4-hour washout, and 5-hour washout conditions are all the same. This would seem to indicate that carboxylone loses its PGP inhibiting effect within 1 hour after being washed out, since both the 5-hour and 4-hour washouts return to baseline. The only condition with significant PGP inhibition is the continuous exposure condition. We see a similar result in the gap junction inhibition graph. On the y-axis here, we have center to outer rim fluorescence ratio. As discussed, a lower ratio indicates more gap junction inhibition, as this means that there is more calcine accumulating on the rim and less reaching the center. In the graph, we see that in the 4-hour and 5-hour washout conditions, there is gap junction inhibition and they are very close to each other, so carboxylone seems to still have a gap junction inhibiting effect even after 1 hour of washout, though it is still lower than the gap junction inhibition in the continuous exposure condition. Here we can see the results for the 24-hour carboxylone treatment. The 4 conditions for this experiment were no drug control, a 24-hour washout, a 24-hour continuous treatment, and a condition in which the drug was washed out after 5 hours of exposure but imaged 24 hours later. Again, we have the PGP inhibition and the gap junction inhibition quantifications. Interestingly, in the PGP inhibition graph, we can see that the 5-hour washout showed significant PGP inhibition and even more inhibition than either 24-hour treatment conditions. In the gap junction graph, we can also see that the 5-hour washout condition shows significant gap junction inhibition. This suggests that carboxylone's inhibiting effects last at least 24 hours after the drug has been washed out, and may be biphasic for PGP inhibition, as the inhibition is initially quick to disappear after washout, but then returns after 24 hours. Here we have the dual assay results for our 5-hour lindane treatment with the same 4 conditions as the 5-hour carboxylone treatment: no drug, 4-hour washout, 5-hour washout, and 5-hour continuous treatment. In the PGP graph, we can see that again the continuous treatment condition has the most PGP inhibition, but what is interesting is that while the 5-hour washout shows the same inhibition as the no drug condition, the 4-hour washout is much higher, suggesting that there may be an inhibiting effect returning after 1 hour. However, as Stephen and Charlotte showed, lindane's PGP inhibiting effect is not very significant when compared to other known PGP inhibitors. Unfortunately, our gap junction results are not very clear. As you can see, there is no significant difference between the inhibition levels of the different conditions. Our 24-hour lindane experiment was also compromised, as after 24 hours the cells had no significant calcine signal and thus may have died. This suggests that the dose of lindane used was highly toxic to the spheroids. However, our results do show that duration of effect is an important consideration when assessing toxicity, as these drugs seem to show an inhibiting effect lasting well beyond the drug's removal from the system.
Hi, I'm Nimesha, and I'm going to give you a background on chimeric spheroids. Chimeric spheroids are spheroids that are made up of two different cell types. The reason why the second half of the HHMI drug discovery team decided to use chimeric spheroids is because they are a better representation of multi-layered human tissue, seeing as how human tissue is three-dimensional and made up of many different cell layers. Another reason why it's a better representation is because it's a really good cancer tumor model. Because cancer tumors have a very high mutation rate, and this means that there's a lot of different cell types in the cancer tumor, and we want to know how the drug will interact with the different cell types. If we add inhibiting drugs to chimeric spheroids, we can learn about the effects on gap junctions and efflux pumps. Some drugs that we used this summer include verapamil, which is a PGP inhibitor, KO-143, which is a BCRP inhibitor, and carbinoxolone, which is a gap junction and PGP inhibitor. As you can see, by using the fluorescence, we learned a lot about inhibition, gap junctions, and efflux pump activity.
Hi, I'm David, and I'll tell you about Lucia and I's project in gap junctions and chimeric spheroids. We're working with two different types of spheroids, KGN cells, which are human ovarian granulosa cancer cell line that do express gap junctions, and MCF7 cells, which are a breast cancer cell line that do not. We had two parts of our experiment this summer. The first part was to observe the self-assembly of these chimeric spheroids, see what pattern they followed. And the second part of our experiment was to investigate how drugs, specifically a model drug, calcine, moved through these chimeric spheroids with the idea that that'll hopefully allow us to more accurately model tumor tissue in future drug testing, toxicity testing experiments. The experimental design for our self-assembly experiments is pretty basic. We just tagged each cell type with a different color of cell tracker dye, seeded, allowed self-assembly, and took a single snapshot of the spheroids, and here are the results. You can see very clearly that KGN self-assembled, self-sorted into the middle of these spheroids. This is true across all ratios and all sizes of spheroid. It is, however, clearer, more pronounced in larger spheroids, 2,000, 3,000 cells per spheroid, and those are the sizes that we used in future experiments. We might attribute this effect, possibly, actually to the presence of gap junctions in KGN cells, because it's known that gap junctions do influence cell-cell adhesion and self-assembly, and the stronger cell-to-cell adhesion forces in KGN cells might be responsible for moving them toward the center of the spheroid and pushing the MCF7 cells to the outside. We did another self-assembly experiment on the effect of cell tracker dye. And in this one, this was sort of a control experiment. We just switched the colors that we were tagging the different cell types with in order to see that it wasn't actually the cell tracker influencing the experiment, that the molecules of dye embedded in the cell membranes were not influencing the sorting. It turns out they weren't. KGN was still moving to the center regardless of what color it was. After this, we moved on to an experiment in drug penetration of the spheroids. We followed a experimental design very similar to other groups, dyeing homogeneously with cell tracker red, eventually adding CBX as a gap junction inhibitor, calcine AM, taking a series of time points. The results for this for MCF7 are what you'd expect. CBX had no impact because it's a gap junction inhibitor, and MCF7 doesn't have any gap junctions to inhibit. The y-axis in this graph shows the fluorescence in the center ratio to the outer rim, so lower implies more calcine in the outer rim, and in this it's the same for both levels of the experiment. That stands in opposition to the trial with KGN, in which CBX showed a strong effect in keeping the calcine in the outer rim, whereas spheroids not treated with CBX allowed a lot of it to penetrate toward the middle. This makes sense again because pure KGN has gap junctions, allows calcine to move. Chimeric spheroid of 50% KGN and 50% MCF7 shows a very small impact from the CBX. We think this makes sense also because MCF7 is, again, on the outside, and so is sort of the first line of defense the spheroid has against the calcine. And so you don't see a strong impact by the CBX because it's inhibiting gap junctions again the MCF7 doesn't have. There's still some calcium that manages to penetrate toward the KGN and the gap junctions though.
Hi, my name is Leo, and as I mentioned earlier, the project that Nimesha and I are working on is to investigate the synergistic effect of ABC transporters in chimeric steroids. Our goals for this project is to investigate and compare the level of inhibition of breast cancer-resistant protein and P-glycoprotein after adding certain inhibitors. And any major differences that we see may reflect synergistic effects of ABC transporters in chimeric steroids. And an example of a synergistic effect that we might see is increased BCRP and PGP activity in a chimeric steroid relative to a non-chimeric steroid. So we mentioned BCRP, but what is that? BCRP stands for breast cancer-resistant protein, and similar to PGP, which was already mentioned, BCRP is an ABC transporter that protects against xenobiotics. However, it is upregulated in tumors, and because of its wide range of substrates that includes many common chemotherapeutic agents, it contributes to multi-drug resistance. Our experimental design followed a very similar pattern to that of the previous groups, except instead of using KGN and MCF7 cells, we use HEK cells, which are short for human embryonic kidney cells. The first transfected cell line that we used was HEK-MDR, which overexpresses PGP, and the second is HEK-R2, which overexpresses BCRP. So first we labeled our cells with cell tracker red and green, and then we seeded these into agarose gels at a density of 900 cells per spheroid. We made 5 gels of HEK-MDR, 5 gels of HEK-R2, and 5 gels of chimeric spheroids. We made our chimeric spheroids by adding a 3:7 ratio of HEK-R2 cells to HEK-MDR cells, and then we waited 24 hours for our spheroids to self-assemble, and then we pre-treated our spheroids with inhibitors, so CoA143 and verapamil. CoA143 is a BCRP inhibitor and verapamil is a PGP inhibitor. And then, depending on the experiment, we added a model drug, either Hoxt 33342 or calcine AM. Hoxt is a BCRP substrate while calcine is a PGP substrate. And then we acquired images over 2.5 hours. First, we took cell tracker fluorescent imaging to determine a sorting pattern of our chimeric spheroids, and then we took images every half hour to create a time lapse to determine the Hoxt or calcine uptake. So we mentioned a 3:7 ratio of HEK-R2 cells to HEK-MDR cells, but where does that come into play? So for our data analysis, we're comparing the uptake of the model drug, so either Hoxt or calcine, between a true chimeric and a mathematical chimeric. Our true chimeric refers to the spheroids that we actually seeded by combining HEK-R2 and HEK-MDR cells. And our mathematical chimeric spheroids refer to a theoretical spheroid that is composed of the 30% of an uptake of the pure HEK-R2 spheroid and 70% of the uptake in pure HEK-MDR spheroid. So these are the results that we've had for our experiment where we use Hoxt as our model drug. The graphs show Hoxt uptake over time for our control spheroids, and then our spheroids that were treated with CoA143 and verapamil. CoA143 is a BCRP inhibitor, and verapamil is a PGP inhibitor. What's interesting to see first is the CoA143 graph. We see that the total Hoxt uptake in the chimeric spheroid is significantly lower than that of the theoretical chimeric spheroid that is composed of 70% MDR and 30% R2 at every single time point. This suggests that BCRP and PGP somehow behave differently in chimeric spheroids than they would in the individual pure spheroids separately. The next interesting point is to look at the verapamil graphs where we see an opposite effect. We see an initial higher rate of Hoxt uptake in the chimeric spheroids relative to the theoretical chimeric spheroids, except this effect wears off over time and the total fluorescent Hoxt uptake by the end of the 150 minutes is equivalent for both chimeric and theoretical chimeric spheroids. We repeated the same experiment except we used calcine as our model drug instead of Hoxt. Except in these experiments, we see that there was no significant differences between the calcine uptake of chimeric spheroids and theoretical chimeric spheroids, which suggests that there was no synergistic effects present for the BCRP and PGP. The greater likelihood of synergistic effects using Hoxt as our model drug may be a reflection of the fact that Hoxt might be a better dual substrate of PGP and BCRP compared to calcine.
We've all had such a great time this summer working in the lab, working with our fellow students and working with the graduate students. From learning basic cell culture technique to making our own posters to present at a symposium, we've had such a great time developing as scientists. We are really grateful for this opportunity.
We would like to thank our professor, Dr. Morgan, our teaching assistants, Beth, Leary, Sean, Curran, and Ben Wilks. We would also like to thank Brown University, the Howard Hughes Medical Institute, the staff at the Multidisciplinary Laboratories, Linda Walsh, and we would also like to thank you, Dr. Carmichael, and the Unilever team for watching our video.
Thank